AP Bio topic 1.1 is on the structure of water and hydrogen bonding. It's a decent place to start the AP Bio curriculum because no life can happen without it. We're all like 70% water, so what's so great about it? Short answer is that it's polar. Every individual water molecule has a slightly negative side and a slightly positive side to it because there's polar covalent bonds between the hydrogen atoms and the oxygen atom. So therefore, each molecule is like a little magnet. And when you have trillions of little magnets bouncing around and flowing everywhere in every drop of water, those magnets attract and repel from one another. And those are hydrogen bonds. And they are what give water its unique properties. Water can stick to itself very well, it sticks to other polar surfaces very well, it takes a lot of energy to evaporate, and it takes a lot of force to break its surface. Water's stickiness allows plants to defy gravity and bring water up from the ground, and it also allows organisms to float on the top of it. Finally, water is really good at dissolving polar substances, which makes it really good for metabolic reactions. AP Biology topic 1.2 is on the elements of life. A big idea in biology is that all living things need to take in nutrients and remove waste in the form of organic molecules. The word organic means from a living thing, but it also means it's carbon-based. Here are the elements that are the most essential to life, but carbon is the most important because it provides the backbone for all organic molecules, and therefore the basis for cells. Carbon atoms are special because they can form up to 4 covalent bonds, which makes them a really good building block for the large, complex molecules that make up living things. Our 4 main groups of biomolecules are carbohydrates, proteins, lipids, and nucleic acids, and here are the elements that make up each. Because carbon is so versatile and can form 4 covalent bonds, some molecules might have the same atoms, but those atoms might be arranged in different ways. Those are called isomers. Geometric isomers have atoms arranged differently around a double bond, while enantiomers are isomers with the central carbon atom that look like mirror images of one another.
Let's talk about topic 1.3, Introduction to Biological Macromolecules. In our last video, we briefly mentioned the 4 main groups of biological molecules. 3 out of 4 of them we refer to as polymers, which are large molecules that are big long chains of individual subunits called monomers. So in other words, if I want to make a polymer, I have to link a lot of monomers into a big long chain. So let's say if I want to make a protein, I have to take multiple amino acids and link them up together. And that is accomplished through dehydration synthesis. So let's say I have 2 amino acids. One has an OH at the end and the other has an H at the end. If I use an enzyme to remove these 2 groups from each other, it's going to form a water molecule and a new covalent bond between those 2 monomers. But what if I want to go from a polymer to a monomer? That is hydrolysis, taking an OH, putting it on one monomer, and an H on the other monomer using a water molecule, and then splitting the covalent bond between those 2. Hydrolysis breaks down, dehydration synthesis builds up. Hey everybody, let's get into topic 1.4. This is about the monomers of all of our 4 groups of biomolecules. The monomer of carbohydrates is called a monosaccharide. Those are usually rings of carbon and oxygen, and they're a major source of cellular energy. When you have 2 rings, it's called a disaccharide, and these rings are linked together by what are called glycosidic linkages. The monomers of proteins are amino acids. All amino acids have some of the same structures. They have an amino group, a carboxyl group, and what's called an R group, or a variable side chain that could be positive, negative, polar, or nonpolar. The properties of that R group or side chain determine a lot of the protein's structure and therefore its function. Amino acids are linked together by what are called peptide bonds. Lipids are a mixed bag, but they're all nonpolar. The monomer of a lipid is what's called a fatty acid. Those are characterized by long chains of carbon and hydrogen. If there's a double bond, the chain is bent, and that's what's called unsaturated. But if there's no double bond and the chain is not bent, then it's saturated. We'll get into nucleotides in 1.6. Topic 1.5, talk about biological polymers. If I take a whole ton of monosaccharides and put them in a really long chain, that's called a polysaccharide. Polysaccharides can either be linear or branched, and they can be used for energy storage or for structure. Linking a bunch of amino acids gives you a polypeptide. Multiple polypeptides can give us a protein. The structure and function of a protein is determined at 4 levels. A protein's primary structure is its sequence of amino acids. Its secondary structure is how those chains fold or coil depending on the hydrogen bonds between the backbones of the amino acids. The shape a polypeptide takes depending on its side chain interactions are called its tertiary structure. These are kind of a combination of the coils and folds from the previous level. If you mess up the interactions between the side chains at the tertiary level, then the protein breaks. Quaternary structure is where polypeptides combine into one functional protein. DNA and RNA, nucleic acids, are made of 1 or 2 really long chains of nucleotides. More details in the next video. Topic 1.6 is all about nucleic acids. DNA and RNA are polynucleotide strands. Therefore, they're made of thousands of connected nucleotides, which is the monomer. There are 5 different kinds of nucleotides. but all nucleotides have a phosphate and a pentose sugar. DNA's sugar is deoxyribose, RNA's sugar is ribose. The third component of every nucleotide is its nitrogenous base. There's A, C, G, T, and U. A and G are called purines because their nitrogenous base has 2 rings. C, T, and U are called pyrimides because their nitrogenous base only has 1 ring. These nucleotides are linked into chains via their sugars and phosphates. In DNA molecules, these 2 long chains of nucleotides meet in the middle and form a ladder shape, with each strand oriented in opposite directions. A's always match up with T's, and C's always match up with G's. RNA only has 1 strand of nucleotides, and they have U's instead of T's. Hey y'all, let's get into topic 2.1, cell structure, subcellular components. So all life is made of cells, and we can categorize them into 2 groups. Are they compartmentalized, and do they put their DNA in the nucleus, or do they not? But all cells have ribosomes, which are used to make proteins, a plasma membrane, which controls what goes in and out, and cytosol, which is a jelly-like substance that fills in space. Plant and animal cells, which are eukaryotic, have a lot more though. We already talked about the nucleus, it contains the DNA. The rough and smooth ER produce proteins and lipids and then ship them where they need to go using vesicles. The Golgi makes sure those cellular products go to the right place. The mitochondrion is the powerhouse of the cell. Lysosomes and peroxisomes break stuff down, and centrioles are useful for mitosis. Plant cells have all the other stuff I just mentioned, plus a few more exclusive things. Chloroplasts are the site of photosynthesis. All eukaryotes have vacuoles, but plant cells have a central vacuole, which is bigger than the rest of them. And plant cells have a cellulose-based cell wall. Hey everybody, let's get into topic 2.2, which is mostly about mitochondria and chloroplasts. A big theme in cells and biology in general is that everything is built to carry out one job. In other words, form meets function. And these 2 organelles are no exception to that. Like eukaryotic cells in general, mitochondria and chloroplasts are also compartmentalized, meaning that different things happen in different places and they're separated out by membranes. And interestingly enough, mitochondria and chloroplasts have 2 membranes. Mitochondria have an outer membrane that's smooth and then an inner membrane that's really folded up. And remember, the mitochondrion's job is to make ATP, and making ATP requires different sets of reactions that happen in different places inside the mitochondrion. The Krebs cycle happens inside the inner membrane in what's called the matrix, and then oxidative phosphorylation, or the electron transport chain, happens along the inner membrane. A chloroplast's job is to produce glucose using sunlight energy. The light reactions happen in the thylakoids, and the Calvin cycle happens in the stroma. Let's get into topic 2.3 on cell size. Another basic rule of living things is that they need to exchange materials with their environment. They need to take in nutrients and get rid of waste. And cells, which are really teeny tiny, have to depend upon diffusion in order to do that. Diffusion is the natural motion of a substance from an area of high concentration to that of a low concentration. Basically, stuff spreads out. Some cells are better specialized for this than others. Cells with a larger surface area to volume ratio, or more surface area per unit of volume, are better equipped to exchange materials with their environment. And if you do the math, you'll see that the larger the cell, the smaller the surface area to volume ratio is. And that's why cells can't be that big. AP Biology asks you to be able to calculate the surface area to volume ratio of a cube and a sphere, and then know which one is better equipped to exchange materials with its environment. Specialized cells increase their surface area through microvilli, like for example, intestinal lining cells. AP Biology topic 2.4 is on plasma membranes. This lays the groundwork for like the next 5 topics, so strap in. You should probably know by now that plasma membranes control what goes in and out of the cell. This means all membranes are selectively permeable. They choose what comes in, what goes out, and what doesn't. And big surprise, the way the membrane is built allows it to be that way. The structure of the plasma membrane is described as being the fluid mosaic model, consisting of a phospholipid bilayer embedded with proteins, glycoproteins, and sterols. The key thing here, though, is the phospholipid bilayer. It's 2 layers of molecules called phospholipids. And lipids are nonpolar, right? Well, these ones are special because they're amphipathic. One side, or the head, is polar, while the other side, the tails, are nonpolar. So then the bilayer is arranged like this, where the nonpolar fatty acid tails face the middle and the polar regions face out. Membrane fluidity is influenced by the saturation of the fatty acid tails. 2.5 is on membrane permeability. Okay, so you remember how plasma membranes are selectively permeable? And the nonpolar regions of the phospholipid bilayer face the middle? And another word for nonpolar is hydrophobic. So what kind of molecules do you think can or can't pass through the phospholipid bilayer? Who gets let in the most? That would be small, non-polar molecules like oxygen and carbon dioxide. They can make it through that non-polar fatty acid region of the phospholipid bilayer. And polar molecules really can't, unless you're really small like a water molecule, in which case sometimes you can get through. But in general, large molecules, large polar molecules, and charged molecules cannot pass through the bilayer. But when cells need large polar molecules like glucose or charged molecules like sodium ions or chlorine ions, that's what they have the proteins for, which we'll talk about more later. Last thing to know here is that the cell wall doesn't do what the plasma membrane does for plant cells. Its purpose is structure and rigidity. Let's start AP Biology topic 2.6 on membrane transport. As I mentioned before, cells often have to rely upon diffusion to take in nutrients and remove waste. Diffusion moves things from a high concentration to a low concentration. It's kind of like going downhill. Diffusion happens when there's a concentration gradient, meaning that there's more of something on one side of the membrane than there is on the other side of the membrane. So if there's a lot of oxygen on the outside of the cell and not a lot on the inside of the cell, what's going to happen? Well, the oxygen will spread out and more of the oxygen will go into the cell. Since that kind of happens automatically and without energy, we call it passive transport. It's not going to work all the time though. What if we need to move something from a low concentration to a high concentration? Cells can do that with active transport, provided they have a little bit of ATP. It takes some work to go against the gradient, kind of like it takes some work to go uphill. If a cell needs to move big particles, it can use a vesicle. When a cell brings something in with a vesicle, we call it endocytosis. When a cell takes something out with a vesicle, we call it exocytosis. Topic 2.7 is on facilitated diffusion. We already talked about diffusion, so what's facilitated diffusion? Well, it's passive transport, but with proteins involved to make diffusion easier for things that can't go through the bilayer. Like ions require ion channels, and water requires aquaporins. But back to the ions. Ions are charged particles, right? So when you have a concentration gradient of ions, it also causes a difference in charge between the two sides of a membrane. That results in what's called a membrane potential, or a voltage between the two sides of the membrane. Kind of like a battery, how there's a charge difference or a voltage between the positive and the negative side. In a cell, this is what we call an electrochemical gradient. And kind of like a battery, a cell can use this voltage to move stuff in and out. A cell's normal membrane potential, or its resting potential, is regulated by the sodium-potassium pump, which uses ATP to pump out sodium ions and pump in potassium ions. Proton pumps do the same thing, just with hydrogen ions.
Let's get into topic 2.8. So we know that water is important, right? And water diffuses as well, which means just like any other substance, it goes from where there's more of it to where there's less of it. The diffusion of water is called osmosis, and it's super important for living things to regulate so they don't lose too much water or gain too much water. In a solution where there's more water, there's less solute or dissolved substances like sugar or salt. Where there's more solute, there's less water. So the concentration of solutes largely determines whether water will go in or out of a cell. Where there's more solute in a cell, water will flow in. When there's more solute on the outside of the cell, water will flow out. And when solute concentrations are equal, osmosis is balanced. Something to remember is that water follows the solutes, and tonicity is super important for cells. Plant cells like hypotonic solutions so that they can remain turgid. Animal cells like isotonic solutions so they don't shrivel or burst. And nobody likes hypertonic solutions. Let's get into topic 2.9. 2.9 provides an overview of how cells move molecules in or out through their membranes. Passive transport is where molecules naturally move through diffusion from a high concentration to a low concentration. A cell does not have to use energy for passive transport, kind of like going downstream or going downhill. Active transport is when a cell uses energy in the form of ATP to move molecules against a concentration gradient, from a low concentration to a high concentration. This is kind of like going upstream or going uphill. Facilitated diffusion is kind of like passive transport, except that it's used for molecules that cannot pass through the bilayer, and it requires the use of channel proteins. Cells can also use charge differences or membrane potential to move things in or out. Electrogenic pumps can use ATP to move ions, which creates a gradient. Then that gradient can be used to bring in other molecules through co-transport. Finally, two more forms of active transport are endocytosis and exocytosis, which use vesicles from the endomembrane system and the plasma membrane to move large particles in or out. AP Bio topics 2.10 and 2.11 are about compartmentalization. What does that mean? It describes how eukaryotic cells have internal membranes that divide cellular processes and basically keep them separated out, and each set of membranes has a different job, aka organelles. Therefore, eukaryotic cells are compartmentalized while prokaryotic cells are not. How did this come to be? The main explanation for this is endosymbiotic theory. So you know how clownfish and anemone live together and benefit one another? That's basically what we think happened between two prokaryotic cells a couple billion years ago. One large prokaryotic cell engulfed another one via endocytosis, and instead of digesting it, the large cell provided a stable environment, and the small cell made ATP using oxygen and glucose. Sound familiar? This tag team of cells eventually became the first eukaryotic cell, the smaller of which became the mitochondrion. It's believed that the same thing happened with chloroplasts evolving from cyanobacteria. We think this happened because these organelles both have double membranes, their own DNA and ribosomes, and they divide independently. AP Bio 3.1 is on enzyme structure. Unit 3 is all about metabolism, which is all of the reactions that happen within an organism. Life itself can kind of be defined as a network of chemical reactions where atoms are rearranged constantly to form new molecules. This requires every organism to have, use, and obtain energy. Metabolism is best understood through pathways, where one molecule is altered in a series of steps and results in a different molecule. Catabolic pathways break down molecules, and anabolic pathways build them up. So living things rely upon reactions happening where and when they want in order to do anything. This is where enzymes come in. Enzymes are biological catalysts, or proteins that speed up reactions in a living thing. Enzymes act upon a substrate. Enzymes' shapes and charge are specifically built for one substrate. A good example is how lactase enzymes only bind with lactose and thus enable the hydrolysis of lactose. Lactase therefore cannot catalyze any other reaction because the only molecule that has the right shape and charge to bind to it is lactose. Let's get into topic 3.2 on enzyme catalysis. Enzymes are biological catalysts, which means they essentially enable reactions for living things. Biological reactions will happen on their own eventually, but not at the rate or when and where living things specifically need them. Enzymes make it so that their specific substrates require less energy to contort and become something else. In other words, enzymes lower activation energy. And when you're a living thing and you're trying to make sure that you don't expend more energy than you take in, saving energy becomes super important. Substrates bind to an enzyme by its active site, which then enables the substrate or the reactant to become a product. The product then detaches from the enzyme, and the enzyme can be used again. Continuing from the last video, lactose binds to the active site of lactase, which lowers the activation energy to subsequently break down or hydrolyze lactose, a disaccharide, into glucose and galactose, which are monosaccharides. AP Bio 3.3 is all about different factors that affect how well enzymes work. First of all, different enzymes have different pH and temperatures at which they work optimally or the most efficiently. Too low or too high of a temperature or pH can drastically change how fast enzymes make reactions happen. For example, enzymes in your stomach, where it's acidic, are not going to function in the intestines where it's more neutral. A higher temperature generally increases reaction rate, but since enzymes are proteins, they can denature or break at temperatures that are too high or pHs that are too low. Increasing concentrations of enzymes and substrates will increase reaction rate up to a point, depending on how much substrate there is for enzymes to bind to. Too much substrate and not enough enzymes will limit how fast reactions happen, and vice versa. Inhibitors are molecules that prevent an enzyme from functioning, which is a way to regulate reactions. Competitive inhibitors will block the substrate from binding to the active site of an enzyme, while allosteric inhibitors will bind elsewhere on an enzyme and change the shape of the active site so the substrate can't bind. AP Bio 3.4 is on cellular energy. By now, we hopefully know that living things are dependent upon chemical reactions, and that these reactions require living things to take in more energy than they use. Therefore, we have to be efficient with our metabolism, especially because energy cannot be reused or recycled. It just gets converted from one form to the next and passed on or lost as heat. One way to ensure energy is being used efficiently is through energy coupling, where in a metabolic pathway, the net release of energy from one reaction is used to drive another. An exergonic reaction causes one of these releases of energy, while an endergonic reaction is one that requires extra energy to begin, and it absorbs that energy from its surroundings. In a metabolic pathway, one molecule gets converted to a product, which becomes the reactant of another reaction. If an exergonic reaction comes before an endergonic reaction in a pathway, the energy from the first reaction can be used to power the second, which wastes very little energy. This can be found all over the map of metabolism in living things. AP Bio 3.5 is on photosynthesis, which is a huge topic. It's the process by which plants and algae convert sunlight energy into chemical bonds in glucose molecules, and also releases oxygen gas into the atmosphere. This process starts with the light-dependent reactions, where chlorophyll and protein complexes called photosystems, located on the thylakoid membranes and chloroplasts, use a photon of light to split a water molecule, separating the hydrogen atoms from the oxygen atom. This releases a high-energy electron, which is passed along proteins in the thylakoid membrane, setting up a proton gradient to make ATP. That electron absorbs a second photon, and that energy is used to make NADPH. The second phase of photosynthesis is the Calvin cycle. This process takes individual carbon dioxide molecules through fixation, then uses enzymes, the energy from ATP, and the reducing power from NADPH to take those carbon dioxide molecules and basically assemble them into glucose, C6H12O6. This is how plants turn light into sugar and power whole ecosystems through this process. 3.6 is on cellular respiration. Respiration is the process by which cells produce ATP, the cellular energy molecule. All life needs ATP, but not all life has to breathe to get it. Respiration can be aerobic or anaerobic, meaning that a lot of cells can make their ATP without oxygen and even switch back to using oxygen when it becomes available. For all cells, however, phosphorylating ADP into ATP begins with glycolysis, where glucose is broken down into two pyruvate molecules, which subsequently produces these products. For prokaryotes, the process stops here, but if you have the powerhouse and you have some oxygen, pyruvate gets converted into acetyl-CoA and entered into the Krebs cycle. This cycle produces these as a result. The NADH and the FADH2 molecules are electron carriers that initiate the electron transport chain, where electrons get passed around to proteins along the inner mitochondrial membrane to produce a proton gradient, which ATP synthase consequently uses to add a phosphate onto ADP to make ATP. Oxygen accepts these electrons, and we ended up with over 30 ATP molecules, which makes it super efficient. AP Biology topic 3.7 is on fitness, and I don't mean making gains at the gym. Another big theme in biology is that variation improves fitness, meaning that the more variety living systems have, the better. This idea is going to come up a bunch more times in this class. The main thing for today, however, is that the more kinds of molecules that cells have, the better they are at responding to changes in their environment. That, by extension, gives them a greater chance of surviving and reproducing in different environments. A big example is that a wide variety of enzymes enables a wide variety of reactions. Even variations in the same kinds of molecules improves an organism's odds. Different chlorophylls absorb different wavelengths of light. Different phospholipids alter the fluidity of membranes. Different hemoglobins absorb and carry oxygen at different developmental stages. You can find examples like this all over the place throughout biology, and they all highlight the fact that the more molecular variation cells have, the better. AP Bio 4.1 is on cell communication. If you're an organism that's made of more than one cell, or you live amongst other cells, then you or your cells must be able to communicate and respond to signals. Cells communicate largely through chemicals, and when something is wrong with the transmission, reception, or response to the signals, you get issues. This is what the first part of Unit 4 is all about. So, how do cells talk to one another? One way is by direct contact, where one cell's surface proteins, called antigens, interact with another's. This is what your immune system mainly uses. The main way cells communicate is by releasing chemicals into their environment, which are picked up by other cells. Paracrine signaling is when the receiving cells are close by, endocrine signaling is when the receiving cells are far away, and chemical messages called hormones are sent through the blood or the lymph. Autocrine signaling is when a cell picks up its own chemical message. But right now, you are processing this information through synaptic signaling, where a neuron transfers electrical impulses from one to another or to a target cell through neurotransmitter molecules like serotonin or dopamine. 4.2 is the introduction to signal transduction. Cell communication can be broken down into three main stages. The first is reception, where a messenger molecule called a ligand binds to a protein in a cell's plasma membrane called a receptor. They are made specifically for one another like a lock and a key. When these two bind, it causes a change in the shape of the receptor, which initiates the second stage, transduction. Transduction can get complicated, but it basically takes the message from the outside of the cell and brings it inside so that the cell can produce the proper response to the signal. This works like a relay race where proteins called kinases phosphorylate or turn on others, which causes the phosphorylation of others, and so on until the right protein or enzyme is activated to produce the response. This is called a phosphorylation cascade. Phosphatases do the opposite of kinases or turn off certain proteins. When a lot of responses are needed at the same time, second messengers are used, which are mass-produced molecules that can amplify a signal and activate a lot of proteins at the same time. The third stage is the response itself, which could make the cell grow, secrete certain molecules, produce proteins through gene expression, or just die. 4.3 is on signal transduction, where we'll get into a few more specifics about transduction pathways. A few types of receptors it'll be helpful to know are ligand-gated ion channels, which do exactly what their name suggests. and intracellular receptors, which are those located inside a cell and responsible for initiating a gene expression response. An important pathway to be familiar with for AP Bio uses a G protein coupled receptor, or GPCR. Epinephrine is a well-known hormone that produces the fight or flight response. When your body releases epinephrine, it prepares you for something potentially perceived to be life-threatening. Epinephrine receptors are GPCRs, so when epinephrine binds, it causes the activation of what's called a G protein, which in turn activates an enzyme called adenylyl cyclase. This enzyme produces a ton of cyclic AMP, a second messenger. CAMP then phosphorylates a lot of different proteins, initiating phosphorylation cascades that will ultimately produce the fight or flight response. One of these results is the hydrolysis of glycogen into glucose through glycogen phosphorylase. There's a lot more examples of this kind of stuff, so please watch the full 4.3 video on my channel. Topic 4.4 is on changes in signal transduction pathways. Our last two topics were about how a cell receives a signal, then sends it to the right part of the cell to produce the appropriate response to the message. There are a lot of moving parts in this process, and therefore a lot of places where this could go wrong. Mutations, which will come up a lot in this class, can alter the shape and thereby the function of a receptor, enzyme, kinase, phosphatase, or any other protein. And that can result in a cell over-responding to one signal and not responding at all to another. For example, P53 is what's known as a tumor suppressor protein, and when it's mutated, it cannot stop a cell's response to a signal telling it to divide, which leads to cancer. Or if insulin receptors are mutated, a cell can't store glucose from the bloodstream, and that leads to diabetes. The other thing that can mess up signal transduction pathways are certain chemicals like medicines or poisons. These could block a receptor, inhibit a kinase or enzyme, among a whole bunch of other things. Some poisons are really good at shutting down key enzymes like adenylyl cyclase or ion channels or pumps, and that can have huge consequences on cell functions, for better or for worse. Topic 4.5 is on feedback. You've probably heard this word before in school. If you get positive feedback on an assignment, you're doing things right. And if you get negative feedback on an assignment, there's something that you're doing that you have to fix. Your cells give feedback to one another on whether or not a change in your internal environment should continue or if it should be stopped. Keeping this internal balance is called maintaining homeostasis, and all living things do this through feedback loops. Here's an example. If you get cold, your body detects a deviation from your normal body temperature, and you shiver, among other things, to bring your temperature back up. If you're too hot, you'll sweat to keep your temperature down. These are called negative feedback loops. In all types of feedback loops, a sensor detects a change in the internal environment, a control center establishes a set point where a certain factor should be at, for example, blood sugar or temperature, and an effector gets told what to do by the control center in response to the initial change in the internal environment. A positive feedback loop will accelerate a physiological change in order to get something done fast. Cells respond to a change in such a way that causes more of the change, which creates a cycle. And that's kind of how babies are born. 4.6 is on the cell cycle. In order for multicellular organisms to grow or repair tissues or continue existing, cells need to divide, and this is their reproductive cycle. Eukaryotic cells spend most of their lives in interphase, which can be broken up into three parts. Gap 1 is where a cell carries out its normal functions and prepares for DNA replication. S is where it copies its DNA, and gap 2 is basically where a cell gets ready to divide into two copies of itself. M stands for mitosis, the division of the nucleus. DNA has been copied, and it needs to be split evenly between two daughter cells so that they are identical. Prophase, the first phase of mitosis, is when the DNA gets condensed and packaged into chromosomes. The centrosomes form the mitotic spindle, and the chromosomes get moved towards the middle of the cell to begin metaphase. Once all the chromosomes are properly aligned and the spindle is attached to the centromeres of all the chromosomes, anaphase begins and splits apart the chromosomes, which elongates the cell. This is followed by telophase, where the new nuclei are forming and the spindle gets broken down. Cytokinesis happens during and after telophase when all the other contents of the cell are divided. The cycle then begins again. 4.7 is on the regulation of the cell cycle. Cell division is a good thing, generally speaking, but too much of a good thing is a bad thing. The rate at which cells divide needs to be controlled. Not enough division means no growth or tissue repair, but too much division is also very bad. A cell's main way of controlling when it progresses through the cell cycle is through checkpoints. Basically, as a cell is preparing to divide, it needs to meet certain requirements before progressing to the next phase of the cell cycle. The proteins involved in these transduction pathways are called cyclins and cyclin-dependent kinases. For example, one type of cyclin activates the enzymes responsible for DNA replication. If something happens to one of the genes for a protein that helps continue or stop the progression of the cell cycle, then cell division can become uncontrolled. When this happens, a cell will typically receive a signal to self-destruct, which is called apoptosis. But when a cell can't properly respond to the apoptosis signal, it results in cells dividing rapidly and uncontrollably, which is cancer. A clump of cells that can't control how much they divide is called a tumor. We call it benign if it stays in one place, and we call it malignant if it finds a way to metastasize or move to another place in the body. 5.1 is on meiosis. Nothing lives forever, and therefore all living things must pass down their genes, which are their molecular instructions to their offspring. Heredity is the study of how this happens. Mitosis is an example of asexual reproduction where one cell simply copies its DNA and splits into two clones. Multicellular life, in contrast, produce offspring that have unique combinations of genes or variation from two parents, which comes from sexual reproduction. Special cells called gametes or sex cells transmit a unique set of genes from each parent and then fuse to form a new organism with a brand new combination of genes. Meiosis is the type of cell division that shuffles the genes and chromosomes each parent passes down, producing 4 haploid cells, aka gametes, which have only one set of chromosomes each. When 2 haploid cells like a sperm and egg cell combine through fertilization, it forms a new diploid cell with 2 sets of chromosomes. In humans, meiosis produces sperm and egg cells with 23 chromosomes each. When the sperm and egg fuse, the future baby will have 23 homologous pairs of chromosomes, 46 in total, with never before seen combinations of genes, which means you are unique and special. 5.2 is about why everyone is genetically different. Meiosis produces 4 non-identical haploid gametes, so 2 divisions need to happen. The homologous pairs, or the pairs in which one chromosome was inherited from each parent, are split first during meiosis 1, and then the sister chromatids, or copies of each chromosome, are split during meiosis 2. Meiosis 1 begins with 1 diploid cell with duplicated chromosomes and ends with 2 haploid cells with duplicated chromosomes. Meiosis 2 thus ends with 4 haploid cells with unduplicated chromosomes, or basically just one set. Just like mitosis, both meiosis 1 and meiosis 2 have the same general phases. Meiosis ensures genetic variation through 2 main things. One is independent assortment, which basically means that homologous chromosomes are randomly divided between the 4 gametes so long as each gamete has at least 1 of each chromosome. This means that each gamete can get any random combination of the parental chromosomes. Second is crossing over, where homologous chromosomes basically swap genes with each other by touching, thereby increasing the number of combination of genes on each chromosome. These 2 factors make sure everybody's different from everybody else.
5.3 is on Mendelian genetics. Genetics is basically using heredity to analyze the inheritance of traits and make predictions. Gregor Mendel discovered that the inheritance of traits can be predicted using laws. He crossed pea plants in his garden, and it basically showed that some traits get masked behind others. Although Mendel didn't know what DNA was, he found out that these masked traits were produced by the inheritance of 2 recessive alleles, one from each parent. And thus, the trait that did the masking and came up more frequently in the offspring was the one that only required 1 allele to be expressed, which is what we call dominant. Alleles are simply versions of the same gene. The alleles an organism inherits are called its genotype, and the genotype determines an organism's phenotype, or its physical traits. Punnett squares use Mendel's laws to predict the outcome of a genetic cross, given the genotypes of the parent organisms. The ratio of offspring produced by 2 heterozygotes, or organisms with 2 different alleles at a gene locus, will be 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive, resulting in a phenotypic ratio of 3 to 1. 5.4 is all about the exceptions to the laws of inheritance that Mendel established with his experiments. Let's start from the top. Sometimes 2 alleles for a characteristic are both dominant. Other times, neither are. Sometimes 1 gene affects multiple phenotypes. Other times, multiple genes impact only 1 phenotype. Sometimes the inheritance of 1 gene at one locus alters the phenotype at another. Some genes aren't even part of the nuclear DNA, but are inherited through mitochondrial DNA. Sometimes genes in question are located on a sex chromosome, like X or Y. All of these break the rules of Mendelian genetics, but here's the big one. By doing test crosses for 2 different traits with fruit flies, Thomas Hunt Morgan found that some sets of genes are more likely to be inherited together than separately, leading to the discovery that some genes are located on the same chromosome, or linked. Basically, genes that are closer together on a chromosome are less likely to get flip-flopped during crossing over, and genes that are further apart tend to get swapped. This flies in the face of the law of segregation that Mendel wrote by saying that genes are inherited independently of one another.
Topic 5.5 is on the environmental effects on phenotype. This whole unit has mostly been about the patterns in which an organism's genes impact its physical characteristics, or its phenotype. But obviously not everything about you is predetermined by your genes right when you're born, right? That would be kind of sad. But anyway, the environment plays a large role in phenotypic variations. Phenotype plasticity is the term that we use to describe when a phenotype is altered by the environment an organism lives in, even if it has the same genotype as the same type of organism in a different environment. In humans, things like skin color, height, weight, and intelligence are multifactorial, or all affected by both genetics and the environment. Here's some more obvious examples. The sex of reptiles when they're born is determined by the temperature at which their eggs are incubated. In orchids, the color of the petals is determined by the pH of the soil that the flowers are planted in. Topic 5.6 is on chromosomal inheritance. Earlier in this unit, we discussed how chromosomes are assorted independently during meiosis, and that each sex cell gets its own unique set of chromosomes, so that when it combines with another sex cell, the diploid zygote that arises from that fusion has a completely shuffled deck of genetic information. This is where genetic variation comes from. Studying how chromosomes are inherited has led us to a better understanding of genetic diseases, some of which result from a mutation in a gene located on a chromosome, or from the inheritance of too many or not enough chromosomes. Disorders like these have been better understood once we have determined their pattern of inheritance, like is the mutated allele dominant, recessive, autosomal, or sex-linked. Some disorders are caused by a non-disjunction of chromosomes during meiosis, where chromosomes do not properly separate from one another, leading to one gamete having too many chromosomes and another not having enough. Having an odd number of chromosomes like this is called aneuploidy, and it leads to disorders like Down syndrome or trisomy 21. Topic 6.1 is on DNA and RNA structure. The focus for Unit 6 is gene expression, or the process by which genes, segments of DNA, are used by a cell to make a specific protein to carry out a specific function. DNA is the genetic material, aka the molecular instructions that your cells got from your parents to build the proteins that make up you. DNA basically enables the transfer of information from one generation to the next. Before cell division, DNA is packaged into chromosomes made of chromatin, where the DNA is pretty much tightly wound around proteins called histones. Plasmids are circular pieces of DNA that are not packaged into chromosomes and are copied separately. It took some amazing science for us to figure out the structure of DNA, which is, as you may recall, a double helix comprised of two anti-parallel polynucleotide strands bound together by phosphodiester bonds between the sugars and phosphates of the DNA backbone and by hydrogen bonds between the nitrogenous spaces A and T and C and G. And remember, RNA is one polynucleotide strand with U as a base instead of T. 6.2 is all about how DNA gets copied. This is super important if we want life to continue on planet Earth. DNA replication is semi-conservative, meaning that each DNA molecule consists of one template nucleotide strand and one newly synthesized strand that was built based off of the template strand. To begin, the enzyme helicase moves along the molecule and splits open the two DNA strands, while single-strand binding proteins make sure that they stay open. Topoisomerase makes sure that the DNA strands don't supercoil upstream of the replication fork. When the template strands are exposed, an enzyme called primase places an RNA primer on both DNA template strands, which serves as a starting point for DNA polymerase to synthesize new DNA nucleotides that match up with those on the template strands. Now here's the catch. DNA polymerases can only make new strands in the 5' to 3' direction. This means that since DNA is anti-parallel, one new strand is built in the forward direction, but the other strand needs to be built essentially backwards. So only a piece of the new strand can be built at a time. These Okazaki fragments are sealed together by ligase as DNA replication continues all the way down the length of the entire DNA.
6.3 is on transcription and RNA processing. Gene expression is all about how DNA contains a code that your cells use to synthesize specific proteins. But your cells can't just directly use the DNA to do that, and mRNA, a temporary copy of a specific gene, must be used instead. Transcription is the production of mRNA from DNA, and it's kind of like using a quote to support your argument when you're writing a paper. You won't cite a whole book or an article, but just a piece. RNA polymerase is the enzyme that produces a complementary strand of mRNA based off of a DNA template strand by prying open the double helix and adjoining RNA nucleotides together as it slides down the template strand. It begins at the sequence called the promoter and ends at the terminator. Transcription factors are proteins that control whether RNA polymerase will bind. To make sure that the mRNA makes it safely out of the nucleus and into the ribosome, and so that the right part of the gene is translated, the primary mRNA transcript is processed. A 5' GTP cap and a 3' poly A tail are added, and it's spliced, meaning that the unused or non-coding regions of the mRNA are removed and the coding regions are linked together, kind of like editing a video. 6.4 is on translation, which is pretty much what it sounds like. Once mRNA is processed and transported out of the nucleus, it's brought to a ribosome, which is an RNA and protein complex responsible for decoding the A's, C's, G's, and U's in the mRNA to assemble the correct sequence of amino acids in order to make a functional polypeptide. The mRNA kind of gets fed through the two subunits of a ribosome like putting originals into a copy machine. 3 nucleotides of mRNA, or a codon, bind with the ribosome at a time, while another RNA, called tRNA, uses its complementary anticodon to match up with the mRNA and bring a single amino acid to the ribosome. For example, the codon AUG will match with the anticodon UAC, and the tRNA with that anticodon will transfer the amino acid methionine to the ribosome. The ribosome continues reading the mRNA 3 nucleotides at a time, and tRNAs put together a chain of amino acids based on the mRNA code until a stop codon is reached, at which point the amino acid chain is cut off. You can translate codons into the 20 different amino acids yourself by using a codon chart like
like this one.
A bit more to talk about in AP Bio 6.4. So far, this unit has mostly been about the central dogma of molecular biology, which basically states that all living things have DNA as their genetic material, transcribe individual genes into RNA, and then translate that RNA into a chain of amino acids in order to build a specific protein. Things called viruses are basically just capsules of protein and some DNA or RNA that take control of gene expression and replication in living cells so that they can force a cell to make more viruses, spreading amongst once healthy cells. Viruses are technically not living things because they cannot metabolize or reproduce on their own without basically hijacking an actual living thing. There are obviously a lot of notable viruses, but one group that we're going to be talking about here are retroviruses like HIV. Retroviruses use reverse transcriptase to, you guessed it, reverse transcription and its viral RNA and convert it into DNA, which is then incorporated into a host cell's genome. That way, the virus's genes are now part of the cell's genes, which can stay dormant and get copied with the rest of the cell's genome every time the cell decides to divide.
6.5 is on the regulation of gene expression. An organism's genome contains the instructions to build thousands of different proteins, but no cell needs to express every one of those genes, so it's crucial to control which proteins are being made and which ones are not. Prokaryotes control gene expression through the use of operons, where the transcription of one gene is turned on or off by the presence or absence of a repressor protein, which binds itself to a segment of DNA upstream of a transcribed region called the operator. Molecules called inducers essentially remove the repressor so that RNA polymerase can bind to the promoter and transcribe. Eukaryotes, on the other hand, control gene expression in a number of ways. Modification of histones in chromosomes allows the cell to choose which genes to express on a chromosome. Negative and positive transcription factors control the binding of RNA polymerase. The different ways mRNA can be spliced can also affect which genes are expressed, and translation itself can be blocked with the use of microRNAs, which bind to a processed mRNA molecule and don't allow it to enter a ribosome. Lastly, some cells can just block or break down already finished proteins. 6.6 is on gene expression and cell specialization. If you're watching this, you're probably a human being, which means that you are made of trillions of cells, which, when they work in a group, make up tissues. Groups of tissues make up organs, groups of organs make up organ systems, and a group of organ systems makes up you. Each of those biological levels of organization has a specific job to do. Almost all of the cells in your entire body are going to do one job and one job only until they die. In other words, your cells are highly specialized. For example, a neuron in your brain does a completely different job than a red blood cell. The function of a cell is determined by its regulation of gene expression, or which proteins it does or doesn't make. Each one of your cells has a copy of your DNA, but it's not going to use all of it, just like you're not going to use an entire library's worth of information to write one essay. A stem cell, which is a cell that does not have a job yet, like high school students, receives signals that tell it which combinations of genes to express and by extension, what type of cell to become. A neuron is going to express combination of genes to send impulses, while a red blood cell will express genes that allow it to carry oxygen. 6.7 is on mutations, which are random changes to genetic information that have greatly affected the history of life on Earth. Mutations are the ultimate source of genetic variation, as they count for 100% of all new genes. A change in DNA results in a change in a protein, which leads to a change in a phenotype or a trait. When a single nucleotide is changed, it's called a point mutation, which can alter an mRNA codon so that it gets translated into a different amino acid, or sometimes even a stop codon, thereby affecting the protein structure and function, though other times it doesn't change anything. But when an extra nucleotide or two is inserted or deleted, that's a frameshift mutation. This changes the groups of 3 that a ribosome reads, and therefore every codon downstream of that mutation results in amino acids that are different from what they're supposed to be. Since prokaryotes don't do meiosis and reproduce asexually, mutations are their only source of variation. So when bacteria divide, any mutations they have are passed down to their offspring or vertically, but they can also pass them horizontally with plasmids. Plasmids with mutations can be picked up in a bacterium's environment, transferred directly from one bacterium to another, or even passed along by accident through viruses. 6.8 is on biotechnology, specifically the kind where scientists use techniques and machinery to manipulate how DNA works. The forms of genetic engineering discussed in AP Bio all require the use of restriction enzymes, which are enzymes derived from bacteria that scientists use to cut DNA in very specific locations so that genes can be isolated for our own benefit. Bacterial transformation is when the desired gene, like say the gene that codes for human insulin, is inserted into a plasmid, which is then absorbed into cultured bacteria to be expressed in large quantities. If we want billions of copies of a target gene, polymerase chain reactions can be used to control high-speed DNA replication using a special heat-resistant DNA polymerase. Restriction enzymes can also be used to tell if the DNA from one source is the same as the DNA from another source through gel electrophoresis. This involves treating 2 or more samples of DNA with the same restriction enzymes and then using a machine to basically sort out the cut up pieces of DNA by size to create an image that can be analyzed. This is what's used in DNA fingerprinting. 7.1 is the introduction to natural selection. In my opinion, if you were to take away one idea from biology class, this should be it. Knowledge conquers fear. So here we go. Evolution is the scientific theory that explains why living things are the way they are. The ultimate explanation that groups of living things change over time and that all life is connected in one big family tree ties the whole study of biology together and makes sense of everything that we know about how living things work. Types of life, or species, do in fact change over time and become new species. It's true. We call it a theory not because it's Charles Darwin's random guess as if we were betting on a football game, but because a theory's job is to answer the questions that we have about the world that begin with why. And evolution does that for our questions about living things. It not only explains why all life is so diverse, but also why we're all related to each other. It also explains why living things are so well adapted to their environments. Darwin's contribution to science not only has shown that species change, but his work also provided the main reason for why species change, which is natural selection. 7.2 is on natural selection, which is the main reason why species evolve. Notice how I said species and not organisms. The first thing to know about natural selection is that individual organisms do not evolve. Populations do. Genetic variation amongst members of a species make it so that there's a variety of phenotypes and genetic traits present in a population. Natural selection occurs when not everyone can survive in a particular environment, and those that have the genes which produce traits providing a particular survival advantage are more likely to survive and therefore reproduce and pass down those advantageous genes to their offspring. It's never an even playing field. Those that have an easier time surviving and reproducing have more fitness, as in survival of the fittest. The environment in which a population lives determines which genes and traits are more likely to be passed down than others, hence natural selection. As generations pass and those with the genes giving a leg up on the competition reproduce more frequently, those genes become more common and the composition or the gene pool of that population changes, and it has evolved. 7.3 is on artificial selection. So natural selection is the main reason why species evolve. And that's when the environment selects particular traits to get passed down based on whether it provides even the slightest advantage for survival and reproduction over competing members of a population. The ones that are most likely to reproduce are the ones that survive the easiest in their environment. So how did this thing evolve? That shape will not help anything survive. This is where humans come in. Artificial selection is when traits that are beneficial to people are more likely to get passed down because humans selectively breed the organisms with their desired traits. We essentially control evolution through selecting which organisms reproduce and which don't, and it's shaped how human society has developed over thousands of years. For example, dogs wouldn't exist without people only allowing particular ones to breed based on traits that are preferable to us. Our food wouldn't look anything like what we have today if generations of farmers didn't use seeds from only the best crops in one harvest they plant for the next harvest. You can think of artificial selection as being man-made evolution. 7.4 is on population genetics. Evolution has been shaping the gene pools of populations on Earth for about 3.5 billion years. Big changes, like the evolution of birds from dinosaurs, don't happen suddenly, and it takes stuff like that millions of years to happen. That's macroevolution, but evolution at its simplest is when a gene pool changes from one generation to the next. If how frequently one allele in one generation of a population is different from that of the next generation, that is by definition evolution, or more specifically microevolution. Little changes over a long time become big changes. Microevolution happens for 5 reasons. The first is natural selection. The second is gene flow, where members of a population move in or out of it, taking their genes with them and altering gene pools. The third is mutations, which create new alleles. The fourth is sexual selection, where genes are more likely to get passed down if they provide an advantage for mating. The fifth is genetic drift, where the reduction in size of a population through some random event or the formation of a new smaller population alters the gene pool, and the frequencies of the alleles in that reduced population don't match that of the population when it was big. 7.5 is on Hardy-Weinberg equilibrium, which is basically when none of the 5 things that we spoke about in the last video are happening in a population, and allele frequencies stay the same from one generation to the next, which means it's not evolving. These conditions are unrealistic for an actual population, so why are we learning about it when it doesn't happen? Well, it's because we can use its mathematical principles to determine whether or not a population is evolving at a particular gene locus by comparing expected numbers to observed numbers. These 2 equations are used to calculate expected allele and genotype frequencies of the next generation of a population. P and Q represent allele frequencies, while P squared, 2pq, and Q squared represent genotype frequencies. If the allele and genotype frequencies in an observed population don't match those that were calculated using these 2 formulas, then that means the population is experiencing microevolution. If the expected values do match the observed values, then the population is not evolving at that gene locus and it's at Hardy-Weinberg equilibrium. Check out the full 7.5 video on my channel for some practice problems. 7.6 is on the evidence of evolution, and it's just a taste of some of the observable evidence that we have to know for sure that evolution is really in fact a thing. It's also what we use to figure out a species' phylogeny. The fossil record can tell us a lot about species that used to exist and when they used to exist based on the layers of rock that they've been buried in. These layers kind of make a timeline that show how similar species have changed over millions of years. We can precisely find out how old a fossil is by measuring remaining radioactive particles in that fossil. Biogeography shows us that similar species tend to evolve in the same geographic area. Embryology shows us striking similarities between vertebrate embryos, once again suggesting common ancestry. Comparing the anatomy of vertebrate species indicates similarity in structure but divergence in function. Vestigial structures are remnants of body parts that modern species have that apparently serve no function at all, but were inherited from an ancient species that once did have a use for it. The similarities in the genome between all forms of life most definitely suggest common ancestry, and finally, evolution can be directly observed through phenomena like the rise of antibiotic-resistant bacteria. 7.7 is on common ancestry, which is something that's assumed to be true if in fact species come from other species, which they do. Darwin himself described evolution as being like a big family tree, where species descend from other species just like children descend from their ancestors. You are more closely related to your siblings than you are to your cousins because you share a more recent common ancestor with your siblings than you do with your cousins, your parents. The common ancestor you share with your cousins is your grandparents, which is more distant. Something that we're going to revisit is the fact that all eukaryotes have a common ancestor, or a species from which we all descend. Endosymbiosis, which we talked about in 2.11, explains similarities that all eukaryotes, including protists, animals, plants, and fungi, share, which are membrane-bound organelles, linear chromosomes, and genes with introns. The explanation that a larger prokaryote engulfed a smaller prokaryote and began a mutually beneficial symbiotic relationship, and that those smaller prokaryotes evolved over billions of years to become chloroplasts and mitochondria, checks all the boxes for how eukaryotes came to be. 7.8 is on continuing evolution. Now, what I'm about to tell you is not on the AP Bio exam, but when you Google evolution, all you see is this picture. And I hate it. One reason why is because people misunderstand it and think that apes that still exist today just became humans, which is promoting misinformation. The actual fact is that apes and humans evolved from the same common ancestor within the last few million years. The second reason I hate it is because it kind of suggests that humans are a finished product of evolution, which is also not true. That leads us to the main point of 7.8, which is on the exam, that evolution is continuous and ongoing. So long as environments change on Earth, which they always will, then species and populations will continue to evolve. All the evidence that we have in the fossil record, in studying the genomes of species, and our constant medical battle against pathogens and the diseases that they cause trying to kill us all the time, all point out that evolution shows no signs of stopping whatsoever. In no way, shape, or form is evolution something that just happened in the past. 7.9 is on phylogeny, which is the study of evolutionary relationships between species or groups of species. Just like a family tree, a phylogenetic tree shows how species split off from older ones, just like how descendants kind of split off from their ancestors. It displays evolutionary relationships, and it can show which species have a more distant common ancestor and which ones have a more recent common ancestor. Basically, how related are species to each other and to extinct species. Biologists make these diagrams based off of the evidence that we talked about back in 7.6. The further back you go on the diagram, the further you go back in time. The branch points, like these, represent common ancestors of two or more taxa. Sister taxa are those that are right next to each other and share a recent common ancestor, and a basal taxon is one that diverged through evolution the earliest from the rest of the taxa on the diagram. Another type of phylogenetic tree, called a cladogram, organizes taxa based on shared characteristics and derived characteristics. It's showing evolutionary relationships based on what species have or don't have in common. 7.10 is on speciation, or simply the formation of new species. The main way to define a species for AP Bio is the biological species concept, which states that two organisms are part of the same species if they can produce viable, fertile offspring. Basically, they can have grandkids. If two organisms can't have grandkids, they are not the same species, and they are what we call reproductively isolated. Pre-zygotic barriers might prevent two organisms from forming a zygote, while post-zygotic barriers might prevent the offspring of two organisms from being able to grow or reproduce. Speciation is a form of divergence evolution, where one group over time evolves to become different enough from another group from which it came that the two groups can no longer interbreed. Even though they're in the same area, something like a mating season or behavior separates a population into two. When speciation happens in the same geographic area, it's what we call sympatric speciation, but when a population is physically separated into two or more by some kind of geographic barrier and evolves separately, it's called allopatric speciation. Both add new branches to the tree of life. 7.11 is on extinction, which is the elimination of a species from Earth, the exact opposite of speciation. The reason that we're talking about it is because, as I mentioned before, Earth and life on it has been around for a very long time. So long that we can't even wrap our minds around it. Less adapted species just go extinct, and that's how it's always been. Background extinction is referred to as the normal, steady rate of species going extinct due to good old natural selection. However, there have been 5 instances, and we are currently in a 6th, where extinction rates are much higher than normal. Those are called mass extinctions. All 6 have been caused by major, violent, drastic shifts in global environments that happened faster than what populations were able to adapt to through evolution by natural selection. Mass extinctions cause a drop in biodiversity, which is an umbrella term for the variety of living things. Extinction decreases biodiversity, while speciation increases it. And wouldn't you know it, biodiversity and genetic variation is like the main thing that keeps evolution going and is super important. And we'll talk about it more in the next video. 7.12 is on variation in populations. Hopefully by now you know how natural selection works, but if you don't remember everything, then I'll remind you that it can't happen at all without genetic variation in a population. If all of the members of a population are genetically the same, nobody will be born with more fitness, and therefore no genes are more likely to get passed down than others, which means natural selection and evolution won't happen. And typically this is bad. Why? Because on planet Earth, environments change constantly. It might not happen instantly, but the environments in which species live are in a constant flux, thereby tilting the playing field for survival and reproduction. So if an environment suddenly changes and a population has no genetic variation, then no individuals have a better chance of surviving, and that population is in trouble. It's hard to bring back endangered species because a small population already has very little genetic variation, which is going to make it even harder for them to adapt. Bottom line is something that we've actually heard before. The more variation, the better. This counts not just for populations, but for ecosystems too, which we'll talk more about in Unit 8. 7.13 is on the origin of life on Earth, and spoiler alert, we still don't know for sure how it happened. Scientists are finding more and more evidence to support claims on how it did happen all the time, but we're just going to cover what you might see on the AP Bio exam. There are several competing models that can help explain it, but all of them agree that life began on Earth about 3.5 billion years ago, and that inorganic molecules had to have formed organic molecules, and those organic molecules had to have formed macromolecules, which must have assembled into a cell. The Opper and Haldane hypothesis suggested that early Earth's atmosphere could have provided the means for inorganic molecules to become organic molecules, which was actually supported by chemical experiments. But at the same time, organic molecules could have been brought to Earth by meteorites. Some naturally occurring substrates could have been the catalysts for the joining of organic molecules like amino acids into biomolecules like proteins. For these molecules to become cells, it's either that RNA enabled gene expression and replication, or that random metabolic reactions enabled the formation of complex polynucleotides, and everything else about life came after. 8.1 is on responses to the environment. In order to stay living, all organisms must respond to changes in their environment. This can be done through behavior or through bodily processes. An organism's response to a change can alter behavior in other organisms, and that behavior can be communicated further and passed along. We see this all over the place in the animal kingdom. One animal's signaling behavior, or perhaps a wide variety of physiological signaling mechanisms, can alter the behavior of animals of the same species, or even that of other species that might do them harm. Communication can help animals find food, claim territory, avoid being eaten, find mates, and so much more. But the bottom line is that they use these forms of communication to improve their chances of surviving and reproducing. Therefore, natural selection increases the frequency of communications that provide an advantage, like cooperative behaviors, for example. There are tons and tons of good examples of all this stuff, so check out the full 8.1 video on my YouTube channel for more specifics. 8.2 is on energy flow through ecosystems. All living things need energy, and some need more, depending on whether they use their metabolism to maintain their body temperature, their reproductive strategy, or their body mass. No matter what though, all life needs a net gain of energy for growth and reproduction, or they'll ultimately die. Autotrophs are organisms that get energy from non-living sources like the sun or hydrothermal vents, and heterotrophs are organisms that get energy from organic compounds in other organisms. You know, eating stuff. In a food chain or food web, which represents energy transfers between species in an ecosystem, we can easily see that autotrophs, or in this case producers, ultimately provide the energy for all of the heterotrophs or consumers. Energy transfer is best represented by a trophic pyramid, which shows energy being passed from producers to consumers and then to other consumers in an ecosystem, with 90% less energy available at each level as you go up. Those at the top get less energy, and energy availability directly correlates with population size. In any ecosystem, you have fewer tertiary consumers than you do producers, for example. 8.3 is on population ecology. A population is a group of organisms of the same species that live in the same place at the same time. These individuals interact with each other and their environment. These interactions affect how the size of the population grows and declines, which is what we call population dynamics. Simply put, a population grows if there are more births than deaths, and a population declines if the opposite is true. When a population is not limited by certain constraints, exponential growth occurs, which looks like a J-shaped curve on a graph. In the equation here, R represents the per capita growth rate, or basically how many individuals each member of the population can produce in a given amount of time. Or it's just the growth rate per individual. Exponential growth occurs when R is at the max, and there are no limiting factors that will slow the population growth. In this scenario, the population is growing, and the rate at which the population is growing is also growing. So what does the population growth look like when there are limits, however? That's in the next video. 8.4 is on the effect of density of populations. A population's density is basically how packed together a population is, or how many individuals there are per unit of area. As populations grow, density tends to increase, and therefore resource availability becomes a problem and population growth slows. Density-dependent limiting factors like competition for resources or diseases slow down population growth as the population becomes more dense. Density-independent limiting factors like habitat loss or natural disasters cap population growth no matter what the density is. The growth model, when limiting factors are present, unlike exponential growth, is called logistic growth, which makes an S-shaped curve on the graph. Logistic growth is characterized by a carrying capacity, or the maximum population size an environment can support. The main thing here is that population growth, or R, decreases the closer the population size gets to the carrying capacity, eventually reaching zero as the population size hits carrying capacity. Exceeding the carrying capacity generally causes a drop in the population size, which will eventually level off to the carrying capacity. 8.5 is on community ecology. A community is a level of organization that's defined by multiple populations living in the same area at the same time. The structure of a community is the number of species in it, as well as the relative number of individuals in each of the populations in it. This is represented by Simpson's diversity index, which is calculated with this formula. Once again, biodiversity becomes super important as it's a factor in how healthy a community is, as measured by Simpson's diversity index. The interactions between these populations in a community influence its structure. The populations can help each other or harm each other and affect their respective population dynamics. Mutualism and symbiosis benefit both species involved in an interaction, while predation, herbivory, and parasitism benefit one population while harming the other. Commensalism is when one population has benefited while the other doesn't care. Competition between populations, however, harms both, and niche partitioning is a process where populations have evolved to use limited resources in different ways, reducing competition between them. 8.6 is on biodiversity. As previously mentioned, biodiversity can be simply described as the variety of life, but it takes genetic variation, species diversity, and community interactions with their non-living environment all into account. So once again, the more diversity, the better. Ecosystems with less biodiversity are less resilient and can't easily withstand environmental changes. A strong, healthy ecosystem has a lot of different species, has an abundance of important abiotic factors like nutrients, a lot of producers for energy transfer, and the presence of one or more keystone species. Keystone species are those that play an extra important role in regulating their ecosystem, even when there aren't that many individuals in their population. Top predators like wolves and orcas are really good examples. Removal of a keystone species tends to lead to ecosystem collapse, and you can easily see how this would be true by studying a food web. Taking these species out would throw off the structure of the whole ecosystem. This kind of event tends to be happening a lot these days, and we'll talk about that in the last AP Bio topic. 8.7 is on disruptions to ecosystems. Evolution by natural selection has made sure that species are well adapted to surviving in their particular environments. Random mutations followed by non-random reproduction has made sure that species can survive when environments change. But a highly disrupted ecosystem might alter the environment too fast for some species, and this is especially true in the age of human beings. The introduction of invasive species increases competition between populations in an ecosystem where native species often lose out because the invaders don't have natural predators. Altered environments due to climate change, habitat loss, and monocropping put massive strain on species. Human activity messes with resource availability in a way unlike any other species that has ever existed, which drastically changes environments across the world. That, on top of the normal geological and meteorological activity that normally changes environments, puts ecosystems at risk like never before. We could spend years talking about all the different ways that ecosystems are being destroyed, so I encourage you to find out more.